Ladies and gentlemen, please silence your phones. Our program is about to begin.
Ladies and gentlemen, please take your seats. Our program is about to begin. أيها السيدات والسادة، سيبدأ البرنامج بعد قليل، لذا نرجو منكم التكرم بالجلوس في مقاعدكم.
Ladies and gentlemen, please silence your phones. Our program is about to begin.
Ladies and gentlemen, please take your seats. Our program is about to begin. أيها السيدات والسادة، سيبدأ البرنامج بعد قليل، لذا نرجو منكم التكرم بالجلوس في مقاعدكم.
Ladies and gentlemen, please silence your phones. Our program is about to begin.
Hello, everybody. Good afternoon. I bet you're excited for this lecture, huh? It's the last of the week, and because it is, we have a very special one for you today. It's remarkably interesting, and it will be moderated by Dr. Imad Ghaloozi. Dr. Ghaloozi is a professor of bioscience and the chief operation officer and associate director of Cow Smart Health Initiative. Professor Ghaloozi has a passion for teaching and student training. In addition to establishing novel courses and graduate programs both at McGill University and international institutions, he co-authored the first Canadian edition of Biochemistry, a textbook for undergraduate students published by Nelson Education, Inc. in 2013. Please join me in welcoming Professor Ghaloozi.
Welcome, everybody. Thank you for coming. My understanding is that's the last presentation before the weekend. So it's really a great pleasure to introduce our speaker today. Neil Ward has more than 20-year experience in the genomic and genetic area, specifically the technology part for sequencing. Neil is Vice President and General Manager for PacBio Europe, Middle East, and Africa. He joined PacBio in 2021. Before that, he worked more than 15 years in Illumina. How many of you know PacBio before coming today? Okay, I see a few hands. So I'm not going to present Neil's achievement. You can look at them online. But one thing today, he will talk to us about this technology, new technology that personally I discovered last year. I did not know what PacBio before that. But it is a technology that will allow us to access the dark genome. You know, we have a dark matter in space. There is a dark genome where it was difficult to access with the prior technologies. And Neil will explain to us how the PacBio will allow that and how PacBio opens new worlds and the interface with AI to really mine the genome and understand it better and make new discoveries, basically in the area, in the domain of rare diseases. Without further ado, Neil, please, the floor is yours.
So good afternoon, everyone. It's great to see a packed auditorium. Thank you for joining us at the beginning of your weekend. I really do appreciate that. And I hope in the next 30 minutes, I can give you some interesting aspects on how AI is coming together with genomics. And I think we're at a really exciting time. This whole winter program has really focused on artificial intelligence and the potential that that brings to us. And I think many of us are aware of the revolution in things like ChatGPT and others. But what I want to talk to you about today is a revolution that maybe has gone less well noticed in the general public and what's been occurring in genomics over the last 25 years. Because these two technologies, computing and genetics, really do go hand in hand. And with the powers that we have today, we can actually understand the language of life, the sequence of DNA at scale and costs that are completely unprecedented in human history. So, as previously mentioned, I work for PacBio, or Pacific Biosciences, as we're also known. We are one of a small number of specialist companies that manufacture equipment and reagents for sequencing DNA. As a company, we're based in California, Personally, I'm based in London, and it's a pleasure to be here at KAUST, and my first time on campus. An amazing resource. I think you're all very lucky and privileged to have this fantastic community around you. At PacBio, our mission is to try and take genomics and really enable the promise of genomics to better human health. And I think we're making some great strides in that over the last few years. And to tell you a little bit about that, I think given that we've got a general audience, I wanted to do a little bit of basic history of where genomics has been over the last 20-odd years, 23 years, I guess. So take you back to June 26th of the year 2000. There was a very grand announcement, to much fanfare at the time, a joint press conference with George Bush and on the UK side of things, Tony Blair, the then Prime Minister, where they very proudly announced that we have completed the first draft of the human genome. I think George Bush himself said, "Today we are learning the language in which God created life." Maybe that was a bit of an overstatement. I'm not sure I believe that, but then I think many of us don't always believe everything politicians say. But certainly it was a real milestone for the genetics community, a project that had initiated in 1990, taken more than 10 years to complete, and had cost somewhere in the region of $3 billion to sequence just one genome. And that was a massive multinational initiative that had researchers from the US, from China, from the UK, many other locations around the world, working collectively as part of the Human Genome Consortia. And interestingly, even at this very early stage, the computational power was a really important component of making this project successful. The initial public-funded effort started by taking large DNA molecules, trying to clone those into vectors. So we don't use them anymore, but things like bacterial artificial chromosomes or yeast artificial chromosomes, so we could get large DNA molecules and then sequence sequentially across those to try and assemble those as a complex jigsaw that made the human genome. Partway through that project, there was additional competition. A private company, Celera Corporation, headed up by Craig Venter, took what at that point in time was a revolutionary approach. Instead of trying to get these big DNA molecules into these artificial chromosomes, they took the approach of just smashing the genome up into tiny pieces and utilizing new sequencing technology, capillary electrophoresis as it was then, to sequence small fragments and relied on computation to try and stitch those tiny fragments together, complete that billions of piece jigsaw puzzle with short DNA fragments. So really, right back in the original inception of genomics as a whole, there's been a bit of a battle between do we do long fragments or do we do small fragments, and computation has really influenced that. But as of 2000, I think the human genome was about 90% complete. There were still lots of gaps in there, bits of errors, things that were wrong. The next few years, between then and 2003, a lot more work was undertaken to try and close some of those gaps and painstakingly walk across difficult parts of the genome. And subsequently, the data was published in these joint Nature and Science publications. And at that point in time, the genome was roughly 92% complete. And I say the genome because actually, it's a little bit of a weird hybrid. The original project enrolled a number of volunteers to provide blood samples that the genome could be created from. And most of the genome material that is the reference genome and has been used ever since 2003 with minor modifications comes from a collection of blood donors in Buffalo, New York. So if we think about it, most of genetics is based on taking an African-American individual, roughly 70% of the reference genome is representative of one individual, and then some other bits from some other people that were stitched together in a weird sort of hybrid. And really, for the last 20 years, all of the genetic analysis that has occurred around the world has taken bits of DNA, aligned it to those reference, and then tried to look for differences. And that's been very successful, as alluded to in the initial introduction. There has been a real ground shift in the ability to do genome sequencing at scale at low price points. And that was largely driven by Illumina. I was very privileged to join that company in the very early days of next-generation sequencing, as it was called. And that really drove the declining cost of sequencing that you see on the left-hand side. So from 2007 through to where we are today, the market leader in this space was Illumina with their methodology. And they moved from doing one DNA molecule in one glass tube, which was the old Sanger sequencing method, to taking one DNA molecule, copying that on the surface of a glass slide into like a little cluster, a bit like a bacterial cluster in a plate of agar, and then doing a sequencing reaction on the 1,000 molecules that you generate in that cluster. And a little bit like a child's flick book, they add one base into a complementary strand. It's got a fluorescently labeled terminated nucleotide, so you can add one letter, you shine a laser on it, you take a picture, and then you measure where all those spots are, and then you deblock and repeat the process. And over multiple iterations of pictures that are built up, you can ascertain a sequence that's typically a few hundred letters in length. When I first started in this technology, I think our longest reads were 26 letters long. Lots of teeny tiny fragments. Nowadays, on Illumina's latest platforms, they typically read 150 base pairs in length. And so we still have this challenge of trying to assemble the complex genome, the complex jigsaw that is these fragments of genomes, by taking all of those small sequences and trying to compare them back to the reference genome, back to these kind of hybrid references, and work out which of those sequence reads map into the different locations, which of them are on chromosome 1, which on chromosome 2, etc. And then try and ascertain if we see a difference in our sequencing read, is that a true biological variation, or is that a mistake? Is that an error in the sequencing chemistry? And that's a computationally really difficult challenge to solve. It's one that has largely been solved now, and there are a number of algorithms that have been developed over the last 15 years. And there are modern iterations, such as Google's team. They take a deep learning approach now to do that. They actually convert the sequence information back into images of an alignment, and they use that image information and image analysis software to determine whether there's a true biological variation in that part of the genome or whether it's a sequencing error based on all sorts of image analysis processes. So it's the first example of now where deep learning and AI really is coming together with genomics. But at PacBio, we do things fundamentally different. Instead of getting these teeny tiny fragments of a few hundred base pairs, we have a technology now that allows us to sequence molecules that are 20,000 base pairs at length, but with the same accuracy and fidelity as that previous next-generation sequencing method. So we start by isolating DNA from your blood sample or from a tissue type or a different organism. We stick some circles on the ends of those hairpins that get ligated to that. And then what we are trying to do is take a single DNA molecule and put it onto our chip, which you can see on the bottom left-hand side. This takes a lot of semiconductor industry manufacturing to make. And on that chip, we have tiny, tiny wells that we call zero-mode waveguides. Those are so small that typically we can only fit one DNA molecule within there. And then we do a sequencing reaction at the bottom of there. We bind a polymerase to the bottom of that zero-mode waveguide, and that polymerase will incorporate the adjacent fluorescently labeled nucleotide. As we do that, we excite with a laser, and we take a movie of this process. And we're watching that movie in real time. We take millions of movies, 25 million movies simultaneously, very high definition, across the surface of this chip. And we're measuring the polymerase as it's incorporating the adjacent bases, each time with a certain fluorophore. And the well is so small that actually the light that's emitted after excitation is a longer wavelength than the diameter of the pore. So the light doesn't escape, it stays localized at the bottom of the well. And so there's some really interesting physics if anyone wants to talk about photonics that occur in there. But the advantage of that methodology is that we don't need to do any amplification, and we have a circular molecule. So we can actually get the polymerase to go around that template molecule of 20 kb multiple times. And each time we generate a sequence read that we call subreads on the right-hand side. Typically, we can go around a 20 kb molecule 7 or 8 times, reading the forward strand and reading the reverse strand. And then we have a slightly different computational challenge. We need to take each of those reads, some of which have some errors within there, and generate a consensus from that in a computationally efficient way. Historically, we used to use a hidden Markov model approach for that. But as a company, we've been doing a lot of collaboration with Google recently and their deep learning team. And they've allowed us to iterate on that process. And we now use a methodology that they call deep consensus. This takes the sequence information, it takes a lot of the kinetic information from those movies, how quickly the polymerase is incorporating adjacent bases. What we call the interpulse duration, the gaps between the incorporation of one base and the next base in the sequence. And then other metrics as well. We put that into an encoder-only transformer, And through that methodology, they can then polish that DNA sequence, stitch them back together, and that underlying kind of deep learning algorithm has been allowing us to get much higher accuracy out of our sequencing platforms than was ever previously possible from long reads. And that's really what gives us now this combination of 20,000 bases of read length, but with an accuracy where we have errors in 99.99% accurate sequencing per each of those reads. So we can start to do some interesting biology with that. The chip itself is about this sort of size, very small. That's where the sequencing reaction occurs. The machine that we put it in is larger. It's more like a freezer. And the Kaus team here, and Nicole, who's over on the right-hand side, and Patrick, have one of our latest Revio platforms. So if you're interested, I'm sure they'd be more than happy to show you what that looks like in the lab. But the top half of the machine is essentially a liquid handling robot. And there's a lot of interesting stuff in robotics. That's probably a completely different winter program at some point in time. But the robotics are able to take all of the DNA and load those in the right session. And then the bottom half of the machine is essentially a supercomputer to do that deep consensus learning algorithm to extract the DNA out of it. And through that system, we now have really a state-of-the-art sequencing platform that can generate highly accurate long reads. Unlike all next-generation sequencing platforms, and in fact, all sequencing that has been done historically, we don't do any amplification of those molecules. We don't modify them in any way. We don't do any PCR reactions. So we have completely native molecules where we can understand not just the DNA, A, T, C, and G, but other epigenetic modifications. So 5-methylcytosine, a methylated base in the human genome, is a common methylation state that we can read out by looking at the kinetic signature. And because we have no PCR and no amplification, we also have very, very uniform coverage across the human genome. Other platforms, if you amplify, sometimes the PCR will make some copies more quickly than it will make other copies. And typically, you get variation in depth of coverage on short-read sequences. We don't have any of those problems. So we now have a platform and a system that can do these really high-fidelity genomes at scale and at a price point that's competitive with what historically has been only available via Illumina. So what are people doing with that? Well, one of the first things people did is fix the reference genome that we'd all been using for the last 20 years. And PacBio were really proud to be involved in this particular science publication that came out in 2022 of filling the gaps in the reference genome that had gone before it. So the schematic on the right-hand side is supposed to visually represent one of each of our chromosomes. The white parts were the bits that had been previously sequenced. All the red parts were the bits where we were just guessing before. We put N's because we said we didn't know. We've now filled all of those in. We know exactly what the sequence information is from one end of the telomere all the way to the other end of the telomere. And that is going to allow us much more comprehensive understanding of genomic variation, irrespective of how complex that genomic variation is. It's still a little bit of a weird reference, though. So to do this particular analysis at that point in time with the computational algorithms that are available, the scientists chose to use a haploid genome. Normally, we have 2 copies of each of our chromosomes, one we inherit from our mother and one from our father. This particular publication took a molar pregnancy, something that occurs very rarely, but results in a haploid genome, 2 copies of either the maternal or the paternal genome. And so this actually is representative of a single kind of copy of a maternal genome. So still not really representative of biology, but a big improvement versus where we've been previously. And if we do that, we can now actually look at how accurate short-read sequencing is versus long-read sequencing. It's been known for a number of years that there are parts of the human genome that are dark for short-read sequencing. And that's because in biology, while statistically 150 base pairs should be unique in 3 billion base pairs, it's not. In biology, we reuse lots of modules. Genes are copied. They have paralogs that are copies that are functional. They have pseudogenes that are ancient copies that have ended up somewhere else in the genome that have degraded over time. And with short-read sequencing, often what happens is at 150 base pairs, you can't tell whether that read has come from the real gene or its pseudogene or its paralog. And so the current algorithms just guess. They take a statistical kind of assumption that roughly they should be equally shared and guess where they come from. And that's what we see on the right-hand side. Now in the telomere-to-telomere kind of supplementary information is this particular chart that shows in the dotted orange line there that 150 base pairs roughly 7% of reads, if you're aligning to the telomere-to-telomere genome, will align identically in more than one location. And it's often those parts of the genome where a lot of interesting biology is occurring. At longer read lengths, we're unique once you get past 20 kb. So this is a little bit of data. If you've not seen these types of views before, this is a browser called the Integrated Genome Viewer that the Broad Institute have developed for many, many years, and is commonly used to visualize next-generation sequencing data. So to try and orientate you, all the gray that we see on here are DNA sequencing reads that perfectly match the reference genome. Wherever we see color on there, that's representation of there being a variation in that sequencing read in comparison to the reference. Some of those colors are truly representative of genomic variation in the biological sample being sequenced, and some of those colors are representative of errors in the sequencing data. And at the bottom, you can see Illumina's very latest sequencing platform, the NovaSeq X, that's been used to generate the sequence data from this particular sample. Exactly the same sample has been sequenced with the PacBio Revio data at the top. And then to orientate you, we've got a particular genomic region here on chromosome 6, where there are two genes that are very similar to one another, HSPA1 and A1B, paralogs of one another. They're both suspected to be involved in hearing disorders. But you can see in that particular genomic region, I don't think I've got a laser pointer, but in that particular genomic region, there's a lot of the Illumina reads are shown as white. That's representative of the fact that they could map to either of those locations, and essentially the algorithms have guessed where they should go. And as a result, it's these types of paralogs or pseudogene events that often cause a lot of the false positive variant calls in short-read sequencing data. And if you looked very carefully, you can actually see some of those on HSPA1B, where obviously the aligners have guessed wrong at a certain percentage of the time, and in the middle of that sort of gene, there's a miscall in the sequence data. On the top, the very long, clean, accurate PacBio data, we don't get those misalignments. At 20,000 bases, each of those reads uniquely align into the genome, and so we can get a much cleaner understanding of the biology that's occurring, have a much better likelihood of working out what's a true biological variation versus an error in the sequencing chemistry. And we can also understand what our two haplotypes are, what we've inherited maternally and what we've inherited paternally, because we can see clear patterns of multiple polymorphisms or variants occurring on the same fragments of DNA. So there's a small part in the middle there. But in the interest of time, I'll move on. I think you get the picture. Ultimately, around the world now, as this technology has evolved, a lot of people are evaluating the different platforms and comparing them with one another. The Broad Institute is, I think, well recognized as one of the largest genome centers in the world. And back last year, they did an evaluation of the multiple different technologies for a very large project that they're doing called the All of Us Project, which is attempting to understand genomic diversity across the different ethnic backgrounds that are in North America. They looked at PacBio's hi-fi data. They looked at Oxford Nanopore's long-read sequencing information. They looked at Illumina. We're able to show that the precision and recall, so what's summarized as an F score here, showed that now with our long-read data, PacBio hi-fi achieves the highest precision and recall, 99.87% accurate at recalling all small genomic variations, so single nucleotide base changes or small base changes under 50 base pairs. But that also we performed extremely well in structural variation, something that's been historically very challenging to ascertain correctly with short-read genomes. So the genome often has blocks that are copied multiple times or inverted or changed in other orientations, which are difficult to assess with short reads. Also, there's a group of about 400 genes that are in the dark regions of the genome but are known to be medically important in all sorts of diseases. We performed extremely well there, and now the Broad Institute are instrumenting using PacBio hi-fi sequencing at scale for the All of Us project based on these findings. So we have a great data set, but even with that, we have needed to develop specialist bioinformatics tools to get the very most meaning out of that we possibly can. So we used Google for single nucleotide polymorphisms and indels with their deep variant software. We, by default, can generate these methylation calls out of our software in 28 million sites for free. There's no extra cost of that. We get that as part of the standard sequencing now. And then we've developed increasingly a number of other more specialist tools and pipelines to call copy number and structural variations accurately. We've got algorithms to really look carefully at the complex pseudogenes, so things like SMN1 and SMN2 that can cause spinal muscular atrophy. Those two copies of genes are 99.9% identical in their sequence. They're thousands of base pairs long. Even the PacBio read lengths can struggle to understand those genes correctly, but we can do that with these specialist algorithms that focus in on those, and we can accurately call the deletions that are often causative of spinal muscular atrophy in those diseases. And then we can look at other parts of the genome that have been inaccessible. So things like tandem repeats. There are a number of diseases, Huntington's probably being the most famous, where there are repetitive kind of trinucleotide repeats in the human genome. And sometimes over time and over generations, those repeat lengths can get longer and longer until they typically manifest in Huntington's disease, a horrible disease that can be really neurodegenerative. We can now start to understand those in blood. We can look at how they expand over time as people age. We can look at the changes of methylation of those. And I think there's all sorts of other additional diseases that will actually be caused by those types of repeat expansions that previously had been inaccessible to genomics as a whole. And we can bring all of those together and do what we call phasing. So understand how the structural variants and the SNPs and the indels and these repeat expansions are all either on the maternal haplotypes or paternal haplotypes. Really start to decipher the biology correctly. But it should come with some warning. We're still taking a lot of these tools and aligning back to the one reference genome. And we all know that our genomes are diverse. I only have to look across the audience here to see that there's a huge amount of genomic diversity, even in this room, let alone in the planet. So increasingly, we're trying to encourage the global community not just to compare their DNA sequence to one reference, but to compare their DNA sequence to multiple references that represent genomic diversity. And there's some new bioinformatics tools and algorithms that allow us to do that and generate what we call pan-genomes. So we can, for each individual, generate a true representation of that individual's genome from telomere to telomere. So we no longer need this sort of hybrid approach of the reference genome. We can do that for multiple individuals, and then we can represent that in a string graph. So again, they can look pretty on screen. They're computationally fairly intensive, and I think there's more room for improvements in the bioinformatics algorithms there. But across the human genome, it's common that there are stretches of nucleotides that are identical to all of us. We all share exactly that same sequence. They're typically then punctuated by single nucleotide polymorphisms or other variants, which we can represent in a string graph. We can see here that we have a block in the middle that is all consistent. We then go to one individual has a C at that next base. The other individuals all have a T. Then we go to another block of DNA that's consistent in most of the individuals, but in one of the individuals at the bottom, there's a sort of orange color. The graph can represent a loop, because in that individual, that particular genomic segment is duplicated. And these graph representations now allow us to much better represent the genomic diversity that we have. And with that, we can also combine short-read data with these graph references to better understand genomics as a whole. It comes with some challenges. It's much easier to annotate one single linear reference genome than it is to annotate these graph references. But again, we're working on that, and one of the biggest challenges is in a graph reference, where are your genes? Because if your DNA sequence is represented in a non-linear structure, how do you represent which bits are coding, which ones code for protein? But we're working through tools for that and re-annotating these graph-based references with common and known variation in the human genome. And PacBio, again, is helping there too. So not only can our DNA sequencing read long fragments of DNA, but we can convert our RNA into cDNA molecules and then read those from one end to another. And that already is allowing us to build data sets today that are helping us redefine what the transcriptome is. So we think we've known what the genes are that are expressed in the human genome for a number of years, largely based on short-read sequencing and some statistical guesswork of how the genes are spliced under different conditions and under different tissues and cell types. What we've seen recently, and have now released this big data set called ENCODE 4, is that the reference maybe had one or two transcripts within their different exon usage, and so the little blocks in that blue diagram at the top are to represent that there were two transcripts. What we're seeing in these data sets is there are a lot of other novel ways that the splicing can occur. And there's actually a lot more splicing in different tissue types than was previously recognized. We found 200,000 full-length transcripts, 40% of which had novel uses of exons that had never been seen before. Those will code for novel proteins in many cases. Those novel proteins will have functions that we'd never been able to assess previously. So it's another whole new layer of information that we can gather from these long reads that we couldn't see before. And we can start to build even more complex pictures as well. So we have DNA sequence, we have methylation by default. We can now also start to understand how the DNA is three-dimensionally packaged. The DNA are 3 billion letters. If we were to actually stretch those out linearly, is about as tall as me. It's about 1.8 meters, maybe this sort of length if we had it linearly. But it's not. We fit all of that into one single cell, into every cell in our body. And we do that by wrapping it around histones and other proteins. And the three-dimensional structure has a huge influence in which genes and which parts of the genome are expressed. And we can now start to understand that as well. We've got this clever methodology where we can use a methyltransferase to artificially methylate all of the A bases in the human genome that are in open chromatin. They're not tightly densely mapped together. They're on the edges. They're the loops of bits of genes that are expressed. And then we footprint that. We read that out in our sequencer, the same time we read the A's, T's, C's, and G's and other methylation changes. And then we can graphically represent that. And it's a complex image. I probably don't have time to do it justice today, but that's what we're trying to represent here, that we can now have multiple kind of modalities of different omics data types showing the different haplotypes with one individual, haplotype 1 and haplotype 2. We have a region of decreased methylation, of open chromatin, and on that top haplotype, we can see how we produce a novel transcript on the left-hand side, and we actually now produce 2 different splice variants on the right-hand side, whereas in the haplotype 2, we only have one gene expressed and one particular transcript that's expressed. It's going to allow us to get into a much better understanding of the non-coding regions of the genome and how they influence the transcripts that we produce in different cells and under different conditions. And we believe will help us uncover the causes of some rare diseases, really allow us to triangulate multiple modalities together. So an exciting time on what we can do with the DNA. So I think what I'm hoping over the next few years is now with these accurate genomic maps, we can really start to chart a clearer path forwards in personalized genetics, and the opportunities are huge. The computational challenges are still difficult. For any one individual, we have probably 4, maybe 5 million single nucleotide variants in our genomes. We have maybe 1 million indels or small copy number variants under 50 base pairs. And we have a few tens of thousands of structural variants of bits of genome that are rearranged or duplicated or copied. Those structural variants actually are often clinically important in disease, but have been very difficult to assess properly previously. A lot of the single base pairs that we focused so much of genetic studies on in the last 20 years are actually benign. They don't make any difference. They're in non-coding regions. They don't change the expressions of disease. But indels, especially if they're near coding regions, are much more pathogenic. They often introduce stop codons. They stop a protein from being full length, or they can change that protein sequence much more significantly than a single letter change. So understanding all of the different variant classes is, I think, going to allow us a much better understanding of genetics as a whole. And then we can bring that together with everything else that's going on in machine learning and deep learning as a whole. Once we understand the novel transcripts, we can convert that RNA sequence into the putative DNA sequence. We can feed that into AlphaFold. We can understand how those novel proteins that we can now represent in our genomic data could have a completely different three-dimensional structure, how they bind to DNA, for example, or how they interact with one another. Huge advances, again, from the Google team with their deep learning algorithms there. And I think through this, we'll start to see a lot of opportunities across all aspects of human genomics. Rare disease is an obvious place to start. These families with unknown causes for maybe intellectual disability or other severe diseases. But the drugs that we take, we need to get to a state where we are personalizing the dosage and the types of drugs that we're using. Neurology, I think there's a whole load of areas of tandem repeat biology to still discover. And cancer itself is often dysfunctional genetics, malformed genomes, transplantation, et cetera. Really all aspects of biology, I think, will benefit from an improved understanding of human genetics. But we also need to digitize medical records, healthcare records, phenotype information. Certainly, sadly, in the UK, where I come from, a lot of medical records look like the left-hand side of this chart, paper copies of stuff. That's not going to be useful if we want to really digitize things. We need to move to more digital, both healthcare records and other information, our wearable devices. But there's amazing work going on. In the rare disease situations, we've got groups taking facial dysmorphology, which is often occurring in a number of rare disease situations, and digitizing those phenotypes so we don't have the variation from one practitioner to another. It's a more consistent recognition of particular syndromes. We have oncology, mammograms, rather than having an individual stare at the screen and use their expert, personalized knowledge of identifying a breast tumor. We can do that at scale with artificial intelligence and image detection. I think bringing all of these modalities together will give us a huge number of opportunities to improve all sorts of diseases that we're currently encumbered with. I've talked almost exclusively about human genetics. We shouldn't forget we are just one part of a huge ecosystem. We have millions of viruses, microbes, organisms that live on our skin, in our bodies. They undoubtedly play an important role in biology and susceptibility to disease, to health, to longevity that we don't fully understand yet. The same is true of our environments. We don't understand the soil microbes. We don't understand microbes on plants. We don't even understand the genomes of many plants that we have around the world. And major projects around the world now are starting to try and characterize these effectively with long read. And I think not only do we have the health benefits, I think there are huge numbers of industrial applications that will benefit from better understanding microbes and how we can modify those. Today we do that, and humanity has done that for thousands of years, of making bread, or in other cultures, making alcohol, or I didn't even realize until recently, coffee itself has a brewing process, a fermentation step that can change completely the taste of coffee depending on the microbes that are on the beans as they're semi-fermented. Huge numbers of other industrial applications that I think understanding genomics will help us benefit from. So I'm a little bit over time, but hopefully that roughly 30 minutes presentation has given you an illustration that we're at this amazing opportunity of converting DNA sequence into a digital form, and by sharing that and understanding that more effectively, I think there are huge opportunities for us across all aspects of biology. So I'd like to thank you for your attention. Thank you again for the opportunity to speak today, and I'm happy to take any questions.
Thank you, Neil, for the amazing talk. And wow, this is a technology, it's opening new doors, I think. So we'll open the floor for questions. We have more or less 15 minutes for questions. There are 2 mics. Please go to the mic if...
And there is never a stupid question. If you're
Absolutely.
thinking it, then probably many other people are thinking the same thing.
The mic is there behind you.
Hi, sorry. Thank you for the presentation. And I was actually curious, talking about long read sequencing, what is your thinking about the new technology that has come recently, nanopore? Do you feel scared about it? Do you feel like a competitor, or is just another technology?
Yeah, it's interesting, and Oxford Nanopore are obviously based in the UK, so I've got friends that work for that company. I think they're doing some great work as well. Ultimately, for science, competition is good. So, the innovations that they're doing on their technology is driving PacBio forwards, and vice versa. So I think it's great for science that there are competing long-read sequencing technologies. One of the maybe disadvantages of the last decade or so is that Illumina haven't really had strong competition to drive their innovation, and that's stalled what could otherwise have come. So we welcome that competition. Interestingly, they're not really newer than PacBio. Both companies have been around for roughly the same length of time. I think there's some interesting business aspects of that. PacBio have been kind of recapitalized. We did a fundraising after Illumina attempted to acquire PacBio technology just a few years ago. But yeah, we welcome the competition. I think both technologies are available here in Kaust's lab, and they complement each other really nicely. So even in the the telomere-to-telomere work, often the backbone of that is assembled with PacBio data, but then the Oxford Nanopore is a really useful validation to ensure that the PacBio assembly had worked correctly. And the slightly longer reads that they have, they're less accurate, so they're a bit more noisy, but they do complement each other nicely.
Next question.
Thank you for the excellent presentation. So I have a question about large language models. It is actually an emerging field of study in modern deep learning. And what is your opinion on how large language models can potentially be applied to genomics research?
I'm really excited about that. I mean, I'm not a practitioner of the tools these days, but my formal education was in bioinformatics, and you know, I used to use hidden Markov models. Large language models didn't exist when I was doing my formal training, but you see the progress that tools like ChatGPT have made in understanding and digitizing human knowledge that's available on the internet and how fantastic those tools are already today. And then we think of DNA sequence. It's just a different language. There is absolutely no reason that we can't take that technology of large language models and apply that to genomic sequence in the same way that we apply it to other written text and start to generate understandings from that. But we were talking a little bit off stage before the presentation. My big concern at the moment of deploying really any deep learning, large language models, or other neural network type methodologies on genomics is that we have a horrible bias in the data sets. And we've got to be very careful as we use modern machine learning and artificial intelligence that we don't just solidify biases that are available in society already. The vast majority of genomes that have been sequenced to date have actually come from people that look and sound like me, people of European descent that are white and are probably middle-aged. And that's not representative of genomics as a whole. So the last thing we want to do is train all of our large language models or other tools on Europeans, and then those tools not perform accurately for people of other ethnicities. So I think there's a real imperative now to make sure that genomics better captures the diversity that we have around the world. And not just the genomic information, but the other phenotypes that go with that, so that we can make sure the benefits of genomics really are for everyone and not just for people who look like people that were in UK Biobank or some of the other cohorts that have gone before.
Yes, thank you very much for the talk. As someone in the field, I'm just curious what your take is on the NVIDIA Illumina acquisition. I mean, I was just curious, or with the merger, how does PacBio see itself with respect to that happening?
It's kind of, it's interesting. I think, as I tried to illustrate through the presentation, the computational powers and tools have gone hand in hand with genomics over the last few years. And PacBio worked very closely with NVIDIA. Our DNA sequencing instruments have a whole load of A100 cards within there. And a lot of the deep learning algorithms that we see today being used in genomics run on those platforms. They're not the only vendor in this space, but they're certainly a major one. So I think whether it's NVIDIA, whether that's Google and some of the new tools that they're producing, or even Amazon have some of their own internal programs, I think we will continue to try and work with all of the major players in that computational space, because we need to be on the cutting edge of computation to make sure that we can continue to iterate and bring even more power into the genomics field. It's an interesting and very dynamic landscape at the moment.
Next question.
Thank you, Dr. Ward, for this fascinating talk. As we all know that AI or machine learning has been very important in the analysis of single-cell RNA sequencing data, and I have also known that the company has recently released the MassSeq toolkit for single-cell long-read sequencing. And I would like to ask your opinion, where AI or machine learning can come into play with regard to single-cell long-read sequencing, and what kind of new tasks that you are expecting AI or machine learning can solve. And another question is that we all know that the RNA sequencing has gone from bulk level to single-cell level and recently to the spatial level. I'm asking that how the company is investing in maybe spatial long-read RNA sequencing, and I would like to I'd like to ask if it's possible for you to introduce the future direction of the company on this topic. Thank you very much.
Sure. Yeah, I think the majority of my presentation was about the DNA, but there's a huge amount we need to learn still about RNA, and modern technologies in single-cell space have allowed people to start to better understand at a gene level which genes are expressed in which cells and how they're co-expressed, and now increasingly how genes in different cells that are adjacent to one another in two-dimensional space or maybe even in three-dimensional space interact. It blows my mind about how you're kind of going to bring all of those data types together. Our focus at the moment, working with the various single-cell technology companies, is actually encouraging each of them to improve the length of molecules that they generate. So 10x Genomics is probably the leading platform in that space, although there are many other methodologies that will generate cDNA molecules with the barcodes and spatial tags on them. But at the moment, their polymerases don't generate full-length cDNA molecules. They truncate it because they haven't needed to. They haven't relied on long-read data. So I think there's a lot of potential for us to continue to work with and share our expertise about how to get full-length cDNA molecules, working with the single-cell companies, so that not only can we understand what gene is expressed in one cell next to an adjacent cell, but actually specifically which isoform of that gene is expressed, or which isoforms, because often one cell can express multiple different transcripts or isoforms. And that's going to just give another extra dimensionality to that data. And I think that's where mentally it becomes difficult. You know, people are good at understanding things in 1 or 2 or maybe 3 dimensions, sometimes 4. But when you start to get to, you know, multi-dimensional data, we're going to need, you know, machine learning tools and other computational tools to help us make sense of those and to draw us some nice, pretty pictures that, you know, can fit more easily, at least into my mind, of what's going on from the biology perspective. So that's our main area of focus. We've other areas as well. I've talked almost exclusively about DNA and RNA today. You know, proteins are hugely important as well. You know, the glycosylation patterns that occur on cells. There's all sorts of other omics data types that I think we increasingly need to bring together to get a true sense of biology that's occurring. And we certainly at PacBio want to be more than just a DNA sequencing company. We want to be one that has tools and products that allow you to answer all sorts of different biology questions, not just DNA related, but to give that sort of scale and complexity. So yeah, lots more to come, but often in partnership with the other single-cell technology providers. Thank you.
Thank you for the wonderful talk. My question is related to the human reference genome. So over the past 2 decades, the human reference genome has evolved, and there has been frustration and reluctance in the scientific community to transition from one version to the other. And now the companies are coming up with this concept of pangenome, as you showed. So are we at the end of the road? Like, is it going to saturate at some point in the recent future, or is there more in the box?
There's definitely more in the box, because I think pangenomes are an interesting step forward. But as you say, there are many groups that I visit around the world that are not even on build 38 of the reference genome. Their whole bioinformatics pipelines are on prior revisions because it's been a big lift to move the annotation from one genome build to another without, you know, some errors that creep in through that. So I think, you know, we need to continue to build tools for those liftovers, and those are improving. So we can go from build 37 to build 38 to the telomere to telomere build. I think we'll increasingly need to work out how to most effectively use pangenomes. But I don't think pangenomes are the end. I actually think the way the technology is going, for each genome that we sequence, we can and probably should just de novo assemble it. Now, we don't do that at the moment because a de novo assembly with current computational capabilities is computationally intensive and therefore slow. It would probably slow down all of your big analysis pipelines. But what we're increasingly seeing around the world as we do genome sequencing of one individual versus another, it's not uncommon that, you know, maybe I've got bits of DNA missing that are in the reference, or I may even have genomic features that aren't in the reference, and those ones in particular get thrown out. You know, if I've got a bit of DNA that doesn't map the reference, then it ends up in the bin and isn't used. And certainly, you know, projects in Dubai recently, there was an Arab pangenome published by PacBio customers that showed hundreds of millions of base pairs of genomic material in a relatively small population of Arab samples that weren't in the reference. And I think in cancer and other diseases, you know, ultimately a de novo assembly, an individual personalized representation of my genome, then compared, jigsaw to jigsaw with one another, I think is ultimately where we'll go. But again, the compute needs to catch up with really where the genetics technology is for us to move to that future state.
Thank you.
I think we have another question.
And thank you, by the way, for so many interesting questions.
Hi, thank you for the conference. I wanted to ask, you highlighted through the presentation the importance of a diverse image or representation. So how could you break, I would say, the challenge of sequencing in probably countries or areas that you don't have the resources to sequence or to get like these samples? So how could you overcome the fact that you want a diverse picture, but you don't have the resources to get there?
Yeah.
Do you think it's effort that you should do as a company, that it's an effort that should come from the researchers, from the governments? What is your opinion?
I think it's going to have to involve multiple stakeholders. So there's no simple solution to it. I think there are increasingly philanthropists, funding bodies, and others around the world that are really keen to level up that sort of diversity that there is in the genomic databases. But there's some challenging history as well. For a number of years, it's been recognized that that is a problem. But increasingly, we're seeing around the world concerns around security and privacy of genomic data as well, and how genomic data is shared. So, one could imagine a situation where a lower or middle-income country has a grant from a charitable foundation to help their genomic information. But we're increasingly seeing that country then sees that as its own personal resource and doesn't want to share that data with the world. And the one thing I would hope that we can take from the COVID experience is actually genomic information. It's really important that we share that. For a family with a child with a rare genetic disease, they're probably the only person in that country that has that genomic variation. If we end up with data just siloed within a country or within a hospital even, or within a PI's lab, which is not uncommon, we're not going to get the benefits that genomics can bring. It's only if we can openly share that information with the right privacy protections and controls in place that will get the global benefits. So I think there's lots of challenging components to do that. We are working with all sorts of major kind of funding bodies, and they're early conversations, so I probably shouldn't explicitly name which ones, but there's a growing recognition that we need to do a better job of this. We'll play a part. I'm sure the other technology players will do, the IT providers will do, and governments, charities around the world will try to help. But yeah, if you have any great ideas on how we can solve that problem, we're happy to have suggestions.
Thank you.
Thank you. I was wondering if the prevalence of genetic conditions will be in a rise now that we have more information about the human genome, and if the data that we're going to use in order to predict that, if it's true, will be sort of investigated from now onwards, or if the past data that we have, maybe from medical records that were digitalized, will be useful, even if there was no genetic testing involved, maybe just some knowledge about some genetic risk factors that could maybe be useful for investigators now to sort of delve into those genomes that can now be studied. So do you think that past information is useful, and how can it actually be, or if it wasn't actual genomic information, then it shouldn't be sort of used in a way, it's not as useful?
I think different data sets have different value depending on what questions you're trying to answer. But there is, I think, still a huge amount to learn about humanity as a whole. And I was talking to colleagues of yours earlier about some studies that were recently out looking at ancient fragments of DNA and using genomic information from those to better understand how migratory patterns of cow herders ended up in Northern Europe. Interesting examples. And we have written texts in some situations that describe how humanity has moved around the world, how cultures have changed, how food and diet has changed with those cultures. And we can now sort of validate some of those stories that are still there in humanity with genetic information and with other carbon dating and other tools. So ancient information still has huge value for us in better understanding these things. With regards to disease prevalence changing, humanity doesn't evolve that quickly. You know, I think evolution rates are still fairly constant. We do see different rates of rare genetic disorders in different geographies around the world. There's some really interesting genetics in small island populations where there were maybe a small number of founders that initially discovered that island, stayed there, you know, had children with their children, had grandchildren, etc., and have a lot of kind of what would otherwise be rare genomic variations at a homozygous rate. Iceland is one of the kind of landmark populations that's been studied for genomics for that reason. They've got great historical records. So there's undoubtedly value that we can have from better understanding ancient texts and ancient DNA and other stuff. We're not going to throw that away. It all kind of goes into the mix, and hopefully AI can help us kind of piece it together.
Questions. We are, the time is up, but I will take the two last questions. Yeah.
Thank you for your talk. You spoke about pangenomes as a potential next step for genomic analysis and how it's largely a technological limitation that is preventing these from being used more frequently. In your opinion, what are the major technological obstacles that are preventing the widespread adoption of pangenomics?
Yeah. So generating the pangenomes up until really the last 2 years has been difficult because the long-read sequencing, either PacBio was prohibitively expensive or Oxford Nanopore was maybe a bit too error-prone to get really high-quality telomere assembly. So it's really only in the last 18 months or so that the technology has allowed us to do that at a scale and a price point and a fidelity that have allowed a telomere assembly to be made from a diploid genome. So I think it's just a matter of time that we build more of those telomere-to-telomere assemblies and then try and combine them together as a pangenome. But the algorithms for developing pangenomes, my limited understanding of that, and there's probably people in the room that could correct me, is that there are some challenges that depending on which order you put the genomes in the first place, it can change the graph structure that you get at the bottom, which seems a bit like there's probably more work to do there so that they're consistent. And then the mapping back to the annotation and how we do that in the multidimensional space are probably the current limitations. But there's lots of smart people much smarter than me, thinking how to solve those problems. So I expect we'll see continued dramatic improvement in that over the next 12 months.
Thank you.
Last question.
First of all, thank you for the talk. Just talking about the complex disorders like cancer that is unfortunately changes over time, a clonal evolutional model, and people develop resistance to the treatments. Yes. My question is possible, thanks to the combo of this new sequencing technology with the AI, coupled with, for example, single-cell approach, to predict the outcome of this kind of disease or the response to a treatment? If, for example, a patient given this kind of treatment, it will develop resistance or not?
Great question. And I think a really exciting area of development at the moment, understanding sort of evolutionary biology and how that fits with cancer. It's similar almost to how people did the tracking and evolution of COVID during the pandemic, understanding the variations that are built up. And I think single-cell technology will undoubtedly help there. We can take really small samples. It's still difficult. So single-cell technology has worked pretty well to allow us to get a sort of high-level representation of the RNA, the transcriptome that's expressed. There are still real challenges of getting a whole human genome accurately from one single cell. You have to amplify the material a lot. It produces bias and stuff. So I think there's more work to do to take our technology and others and be able to take just one single cell, one copy of a DNA, not lose any of those molecules and get a full representation of the genome. That's one aspect. I think there's a lot of potential in the data types that we produce or other long-read sequences produce, because in cancer, there's not just DNA changes, there's DNA rearrangements, there are methylation changes and other epigenetic changes that likely are precursors to the DNA damage itself. We can start to try and understand those. But it's going to take years of dedicated scientific work to try and tease these things apart. There's some great projects around the world. I think in London, where I'm from, Charlie Swanton at the Crick Institute there has done some amazing work in high-depth exome sequencing in lung cancer. in the last sort of 5 or 6 years, which has really helped illustrate what are trunk mutations that are consistent in all the cells and which are the different branches and how different treatment types should be combined together. We're probably a bit like in HIV, where one therapy doesn't work. We're going to need combination therapies in cancer, maybe some immunotherapy with another targeted therapy to really make sure that people that often still present with late-stage disease can have more than just a couple of months extension of life, which is typically what happens with most sort of single targeted therapies at the moment. We're going to need to work out how to do that and work out how the pharmacogenomics and everything plays together in that. So I really hope that we can make some big progress in that space over the next year. There's a lot of unmet clinical need, but a lot of exciting and important research to do over the next years. It's going to keep us all busy. You know, if we're interested in genomics, there's plenty of opportunity for you to have exciting careers in this space over the next many decades, I expect. But thank you all for the questions.
Thank you so much, Neil, for the amazing presentation and this discussion. Thank you all for the great questions. And please.
Thank you. Real pleasure. Really helpful.
It's a pleasure. We have a nice gift for you. The souvenir from KAUST.
Oh, wow.
This is really...